Today, we're going to be talking part 2 of the protein purification lecture. This one's going to focus on centrifugation and the different ways in which a centrifuge operates. So there's different types of centrifugation. There's regular centrifugation and there's ultra centrifugation. Regular centrifuge can go up to about 20,000 RPM, depending on the kind of rotor. Some rotors are rated lower, which is about 40,000 G-force. An ultra-centrifuge can go 100,000 RPM, or about 400,000 in terms of its G-force. Now, within these types of centrifuges, there's different types of rotors. And one type of rotor is a swinging bucket rotor. And a swinging bucket rotor, the buckets are actually swinging outwards during the spin as it's rotating around. So you're putting a sample in here and it's rotating outwards to spin. So the greatest force is on the outside of the bucket and the lowest force is toward the inside of the bucket. It depends on the radius. So the force increases as you move outwards in a centrifuge. So this is a swinging bucket rotor. Other types of rotors have a fixed angle to them. And a fixed angle rotor, this one happens to be an SS34 rotor, and it just has, it's angled. So you're putting your samples in at an angle, which means when you see the pellet's going to be right down here. It's not going to be at the very bottom of the tube, like on a swinging bucket rotor. The pellet's going to be right at the bottom of the tube because that's where the greatest force is. In this one, you're putting it at an angle, so you're going to have a little bit of pellet all down the outside of one striped side on the tube with this fixed angle rotor. Other types of fixed angle rotors are vertical rotors. So you can see in this one, you're going to put your sample in completely vertically in here. So it's going to be setting up and running vertically up and down for your sample. Within those types of rotors, there's different types of centrifugation that you can do. You can do differential centrifugation, you can do rate zonal centrifugation, or you can do an isopycnic density gradient. So a differential centrifugation, you're basically separating on the basis of size. The larger something is, the greater the Stokes radius, the Svedberg constant, the larger the size of something is, the faster it's going to move to the bottom of the tube. Compared with rate zonal centrifugation, it's also separating on the basis of size, but in rate zonal centrifugation, it actually uses a density gradient in there only to stabilize your sample, but it's still separating on the basis of size. Now the third type, and we're going to be using all three of these types this semester, the third type is your isopycnic density gradient, and this is a salt gradient that is going to separate only by density. So in this case, it does not matter what size the molecule is. You can have a very large molecule and a very small molecule, but if they have the same density, they're going to separate to the same point in that density gradient. So here's a little bit more about how that works, and specifically something called force factors. If you are using a centrifuge, and let's say you have a centrifuge rotor and you go, crud, the centrifuge rotor that I want to use, I can only spin up to 6,000 RPM. But here I have a protocol which tells me that I need to spin it at 12,000 RPM for 30 minutes. How can I do this procedure? Well, the answer is, if you can't go as fast, you have to spin longer. So you can go for 6 minutes at 1,000,000 G, for example, or if you don't have a rotor that can go 1,000,000 G, because most of them don't, you can go 100,000 G for 60 minutes, 10,000 G for 600 minutes, 1,000 G. You need to separate something on a little tabletop centrifuge that can only get up to 1,000 G. Okay, you can do it, but you have to spin for 6,000 minutes. So there's that, you either need the force or you need the time idea. Now, when you are separating on these different types of gradients, you have, remember I said you have a gradient-free differential centrifugation. So in differential centrifugation, so ignore this for a minute because there's no gradient in differential centrifugation. You're going to load your sample maybe into the top of a tube, and you're going to spin it in the tube. And when you spin it through the tube, it's going to start to separate. The larger pieces are going to separate faster than the smaller sized pieces of proteins or whatever. But as you keep spinning your sample, eventually your sample is going to go to the bottom. Eventually everything is going to go to the bottom as you keep spinning, because it's separating by size. The larger pieces will pellet first, but eventually everything will pellet. Now this is how you can separate organelles, for example, in a cell. If you have a very large organelle, such as a chloroplast, versus a smaller organelle that's in a cell, you can separate them just by having different amounts of time that you're spinning each one. A very short spin is going to bring down the largest fragments and then leave everything else in supernatant, and a longer spin is going to bring down smaller pieces. So if you need to, you can spin for a little bit, get rid of that, a longer period, get rid of that, maybe save the third fraction, or maybe save the second fraction, etc. So you can use differential centrifugation to separate out all the different components of a cell. So the main difference between differential centrifugation and rate zonal centrifugation, as I said, is that it uses a gradient only as a stabilizing factor. So sucrose is in there, a gradient of sucrose is in there as a stabilizing factor. So it uses a velocity density gradient just to sort of slow down the separation, make it take a little bit longer, give you a little bit of a greater chance to separate everything out. Now this is in contrast to an equilibrium density gradient. And an equilibrium density gradient, you have a gradient that's made up of different concentrations, for example, of salt. And you can load a sample in one end of it, and everything will migrate to its characteristic density, and it will stay there. So no matter how long you spin, you could spin for 6 years, and if you took your sample out at the end of 6 years, all of these bands would still be in exactly the same place, because that is their characteristic density. So when you are using a density gradient, everything moves to its place of density and then stops spinning. Some other considerations for why you would want to use one type of centrifugation over another. If you have a differential centrifugation and you have a bottle full of stuff, what you have in your bottle full of stuff is you have all different sizes of particles mixed up. So you have large particles mixed with very small particles. So your large particles and your small particles are all mixed, and you start spinning them. So when you start spinning in a differential centrifugation that's just simply separating on the basis of what size pieces are coming down, you're getting, in the side of that bottle, you're getting some contamination. So first the large pieces come down, but you're going to have some small pieces that are right next to the large pieces when you started spinning. So you're going to get mostly large pieces, but you're going to have some small pieces in there as well, and as a contaminating factor. So you can, in a differential centrifugation, you can have very large amounts, very large bottles. You can spin 1 liter at a time in some bottles, or you can spin 250 mils at a time in some bottles, but they're a little, differential centrifugations a little more prone to contamination. Now, if you are using rate zonal centrifugation, one of the things of rate zonal centrifugation is you're starting your samples up at the top and it's separating out. So you're starting your samples all up at the top, and whether it's fixed angle or whatever type of rotor it is, you've loaded your entire sample up at the top that you want to separate. But the downside of this is, instead of being able to separate maybe 250 mils at a time or 1 liter at a time, you're limited to maybe just doing a couple mils at a time. So you get a much more pure separation with rate zonal centrifugation than differential centrifugation, but it's at the cost of what kind of volume can you do with that. So you can load something onto a rate zonal centrifugation tube, and it's going to separate out, and it'll separate out at whatever angle you have it at. And when you stop spinning it, everything's going to be separated into its characteristic sizes. And this is for a short type of spin. But even if you have it in a vertical rotor, whatever type of centrifugation you're doing, a vertical rotor sets it up exactly the same. So you can load your sample in it and it will separate out, but it's separating the layers vertically. And when you stop spinning it, all those vertical layers then shift, so you still get those same bands in there, even when you're using a vertical rotor. Okay, so now I wanted to talk about why you might want to use different types of either differential rate zonal or a density gradient. So this is sort of a graph of the ways you would use to separate things. So things which have different S values, different sizes, you would want to use to a sedimentation, you'd use a sedimentation coefficient to be able to say, okay, this has a different size. On the other hand, some things might have the same size but a different density. So for example, if you were trying to separate peroxisomes and lysosomes, peroxisomes and lysosomes are about the same size. So you'd have to use a density gradient to separate them because they have different densities. So on the other hand, maybe you wanted to be able to separate chloroplasts from some soluble proteins. Well, chloroplasts and soluble proteins have approximately the same density, but they have very different sizes. Chloroplasts are very much larger than soluble proteins. So if you were doing this, you can separate chloroplasts from soluble proteins by using differential or rate zonal centrifugation to be able to accomplish that separation. So we've talked about a lot of different techniques. A general purification scheme here. You can disrupt your cells, and you can separate proteins from a cell supernatant. At a certain point, when you've blended all your solution up, you're going to have a large volume, and you want to be able to concentrate that protein down. Now, there's different ways of concentrating your protein. You can use something called ultrafiltration. Ultrafiltration, you're putting it through filters which have very, very small holes in it, which will let, aqueous solutions, for example, through, but will not let, proteins of, say, 12 kD and up through. It'll hold all of those back. So you're not really separating anything except really, really large from, say, really small, like aqueous solutions. On the other hand, if you wanted to just be able to precipitate everything, you can use acetone, an organic solvent, to precipitate all of your proteins. It's a good method in some ways, but sometimes proteins have trouble recovering and going back into solution after they've been precipitated with something like acetone. So it can be useful, but sometimes it can hurt your protein. You just sort of have to try. We've used it sometimes in the lab I work in, and other times it didn't work for other proteins. So it's just it's a trial and error kind of thing. You can use salt precipitation, and this is what we're going to be doing in this class in our third experiment. We're going to be using a salt called ammonium sulfate to precipitate out our proteins. And then after you've used salt to precipitate out your proteins, you need to get rid of the salt so it can go back into solution. So there's something called dialysis, and dialysis is putting it into a little bag with tiny holes in it, which will allow the salt to leave the bag and other solutions to flow in, free exchange of solutions because of the small pore size. And you can actually buy dialysis tubing with different pore sizes. You can buy it so that it will let in anything less than 500 Daltons. You can buy it so it's 10,000 Daltons, 50,000 Daltons, and you can use it to get rid of the small proteins if you buy it with 50,000 Dalton pore sizes even. So So you can use this to get rid of small things, or you can use this just as a buffer exchange, or just to get rid of salt, things like that. So by using different size holes in the tubing, you can decide what you're allowing in and out of your tubes. You can also do gel filtration columns to remove the excess salt. So a gel filtration column is going to separate out by size. And because it separates out by size, salt is very much smaller than the proteins. So you can load proteins in the top. Proteins will come out way before any of the salt will. So you just collect the fraction that has the protein that you're trying to get in it. There's chromatographic separation. So we're going to be talking in this class about 3 different types of chromatography. We're going to be learning about different types of chromatography. There's ion exchange chromatography, and that's using a salt exchange. So it's the charged proteins are attracted to the media of the column, and as you add higher concentrations of salt, you will cause different proteins to come off of your column. You can have that gel filtration that I just talked about that separates just on the basis of size. There's no attraction to the column at all. It's just the small things tend to get caught up in the column and allowing large things to go on through. You can do affinity chromatography. In affinity chromatography, you actually have something which is causing it to bind to the column, and then you need to do competition to get it off the column. So when we do our aldolase experiment, we're going to be doing this type of chromatography because we're going to bind our aldolase firmly to a column, and we're going to have to compete it off with the natural substrate for aldolase. You can also, as part of your general purification scheme, after you're through running it on a column, you might want to concentrate it again. If it's pure, you might be ready to lyophilize your protein. And lyophilization is basically freeze drying. You get it very cold, you freeze it down, and you use a lyophilizer to pull off all the liquid so that you have your protein in a nice stable state so that you can just maybe measure out small amounts of your protein. You might want to do ultrafiltration of it. Back what I talked about here, ultrafiltration, allowing particular things to go through. So if you're concentrating it, you're putting all your protein solution in there, you're getting rid of all the excess water, holding back all the proteins. Sometimes if people don't have a lot of money, because lyophilizers are expensive to buy, expensive to maintain, ultrafiltration cells are, again, very expensive to buy. A low cost method is putting your protein solution into a dialysis bag so that solution can go through your dialysis bag, and putting it into a polyethylene glycol powder. And that polyethylene glycol sucks the water, because it's solid material, it wants to be hydrated. It sucks the water right out of your dialysis bag. So you're concentrating your protein in a very low cost manner. But generally people don't use that as much, because generally they do have enough money to buy, one of these other options, or to borrow a lyophilizer or something like that. And eventually you're going to do preparative electrophoresis on it. You want to make sure that your protein is very pure, with an ultimate goal maybe of crystallizing your protein so that you get some more information if you're doing particular types of reactions with it. Okay, so when you are working on your enzyme, there will be a chart that you will fill out, because you want to keep track of your progress, of how your purification is going. So you might say something like, in the beginning, here's what I started with. And let's say in the beginning, your volume was 1,000 mils. Oops, here, 1,000 mils. And let's say you measured it and it was 10 units per mil. So 10 units per mil. So that means your total units are 10,000. And then let's say you did a Bradford assay on it, and your Bradford assay came back and said, okay, your protein is 3 mgs per mil. And so then you want to know, what's the specific activity of my protein? Well, specific activity is units per mil, units per mil, divided by mgs per mil. So units per mil divided by mgs per mil. So your specific activity here is 3.33. Now let's say in the beginning, we're going to call it 100% yield, and your purification level that you start with is 1.0. So then you do a procedure on it. You do differential centrifugation to get soluble proteins. So differential centrifugation. And let's say you magically still have 1,000 mils. And let's say, amazingly enough, you have not lost any units out of there. So you've gotten rid of proteins that you don't want, but you're keeping the proteins that you do want. So you still have 10 units per mil. So you still have 10,000 units. But now you've done a Bradford assay. And remember, the goal of protein purification is getting rid of the proteins that you don't want while keeping the proteins that you do want. So you now have less total protein after you've done differential centrifugation. So now your concentration is 0.1 mg per mil. So now your specific activity is up to 100, but you magically still have 100% yield because you still have those same total units. So you still have 100% yield, but your purification of your protein has gone up 30-fold. You have purified your protein 30-fold by keeping the protein that you want and getting rid of the proteins that you don't want. Now let's say you do another step. Let's say you do column chromatography, and you still magically have 1,000 mils, and it's still magically 10 units per mil, which means you still magically have 10,000 units. But again, now you do a Bradford assay, and now the protein concentration is down to 0.01 mg per mil, which means your specific activity is 1,000. So notice, as your protein concentration drops, and as you are able to keep your units of protein, or even if your units of protein are dropping slightly, as long as your protein concentration is dropping faster than you're losing units, you're going to be increasing your specific activity of your protein. So as you purify your protein, your specific activity is going up. You still have 100% yield of your protein because you still have 10,000 total units, but now you've purified your protein 300-fold here. You can even look at individual steps. You can say, okay, between where I began and differential centrifugation, I have purified my protein 30-fold. And then between differential centrifugation and column chromatography, I've only gone up 10-fold. So this was a more efficient step than this one, but maybe this one was necessary to, be able to get rid of most of that last protein that you couldn't get rid of any other way. So you can start making judgments like that on protein purification steps, and you can try out different steps. So when in lab, when we're working with protein, we will sit there and say, okay, well, does this column work? Does that column work better? Which column works best? So we'll try out different columns, and based on the results that we get from here, how much activity are we losing versus how much non-wanted protein are we using? How are we able to increase our specific activity? Then we can choose the next best column for that particular step. So you can do this as you go along, as you're working out of protein purification. Now, as an undergrad, you're probably very lucky working in lab that someone has worked out many protocols for you. But if you go to grad school, then the burden falls on you to be able to say, okay, here I have a protein that's my goal to be able to purify. How am I going to purify? What steps do I need to do to purify this? And you're going to have to try a whole bunch of different things, and you're going to have to keep track of it in a table that looks a lot like this.